Hi, good afternoon. It's good to see you all. We're going to give it just one more minute with the waiting room filling in, and then we'll get started. Great. I think we've got a pretty full audience. Thank you all for joining. I'm going to go ahead and get started on the presentation here. I really appreciate you making the time to learn more about fluid form bio. I plan to talk for about 12 to 15 minutes, and then we'll move into a Q&A section. There should be a Q&A function on the screen in front of you there. So as you have any questions, please feel free to add them into the queue. For the Q&A, we'll try to cover as many of them as we can. We do plan to wrap this up right about 4:30. So I will start by sharing this presentation here. I'm excited to walk you through a quick overview, both of the treatment of type 1 diabetes, what we're doing that's so different, and why we're so excited about where this can take us. I'll start by just sharing a little bit of background on type 1 diabetes. Some of you may be very familiar with this, others this may be fairly new to you. So I'd just like to walk through a little bit on what the experience of someone with type 1 diabetes is like in their day-to-day life. As you may be aware, somebody with type 1 diabetes is unable to produce insulin naturally the same way a healthy pancreas might. Insulin is what's responsible for regulating your blood sugar levels. So without the ability to naturally produce insulin, your body will have wild fluctuations in the blood sugar level, and that can be very, very dangerous. Therefore, when you have type 1 diabetes, you need to constantly monitor your blood sugar levels, and you need to intervene with the management of insulin, either by shots or by a pump, all day, every day, for the rest of your life. Today, there are no cures, there are no long-term solutions. The best solutions we have available to us today include a continuous glucose monitor and the wearing of an insulin pump, which still requires a significant number of thoughts, decisions, and actions every single day just to make sure that your body is behaving the way somebody with a normal, healthy pancreas would. In fact, there's some great literature that's been done on this that show that people with type 1 diabetes actually make 180 additional decisions every single day above and beyond the decisions that most of us take for granted in making in normal life. That includes everything from what and when to eat and what food to bring with you in order to make sure that if your blood sugar goes low, you can respond to it. It includes making sure you have your insulin supplies, you have backups for those insulin supplies, you have test strips for your blood glucose meter in case you need to do a finger prick. It means you have a backup for your continuous glucose monitor, where you're going to go during the course of the day and what that might mean if you need to take some of those supplies with you, and what kind of clothes you might wear if you're going to be needing to give yourself a shot using an insulin pen or if you're going to be wearing a pump. These kinds of decisions take an enormous toll on people with type 1 diabetes every single day. Now, for background, type 1 diabetes, substantially different from type 2 diabetes, which we hear a lot more about today. Type 2 diabetes is your pancreas still produces insulin, but your body becomes less sensitive to it. Type 1 diabetes, your body stops producing insulin, and that's due to an autoimmune response. And so for people with type 1 diabetes, this is not something that is their fault. It is not because they ate something or because they behaved in a certain way. This is an autoimmune disorder with an unknown cause. And it's important to note that for a long time now, people have been trying to figure out how could we possibly cure type 1 diabetes. One of the areas that's gotten a lot of attention for a very long time has been trying to replace the cells that were killed by that autoimmune response with healthy cells that do the same job, i.e. cells from a deceased donor pancreas or cells from an animal or another source that might produce the insulin that they need, how could we transplant those cells? Now, this work's actually been going on for longer than my lifetime. The first islet cell replacement ever done in a mouse was in the early 70s. And the first islet cell that was ever done in a human, the replacement treatment, was done in 1980. In those early days, we saw a very, very low success rate, but it was just encouraging enough for scientists to keep chipping away and keep working at it through the 80s and the 90s that led to the development of what became known as the Edmonton Protocol. At this time, we had developed better immune suppression drugs and regimes, and that allowed researchers to then develop a standardized protocol that didn't rely on steroids in order to start expanding the research and work in this. In that world, we still saw about a 10% success rate, but that was at 5 years, not after 1 year. So it was an improvement in that direction. And in the 2000s, that was when we also tended to see the first real optimism that maybe there's something here. We are seeing cells that can do the job. Perhaps one day this could be a cure for patients. As a result of that optimism, the decade of the 2010s was a decade of enormous innovation rushing in. This included the Nobel Prize for induced pluripotent stem cells, and then the development of protocols around how to turn those stem cells into islets. It also included a lot of work around cell encapsulation and different ways to protect cells from the immune system. This first wave of innovation helped improve the cell supply because now we could rely on stem cells, not just deceased donors. And it really developed the first testing out of this notion: what if we could protect these cells from the immune system without having to suppress the immune system for the whole patient? And the last 5 years have been just as exciting. We've seen the first clinical trials of the innovative products that were starting to be developed in the 2010s with Vertex and their trial. We've seen the approval of a deceased donor-based islet cell treatment that's now on the market today. It's called Lantidra. And what we're seeing is all of these early generation products still require systemic immune suppression. We haven't solved that yet, but we're now starting to march towards a direction where we're seeing promise. We're seeing excitement and enthusiasm. We're seeing enormous investment. That includes large pharma companies like Vertex, Novo Nordisk, Eli Lilly, and others. What's really interesting is when you look back to the early days, the major challenges that existed then generally fell into 3 buckets. Failed engraftment, meaning did the cell find enough blood supply to survive, thrive, and function? A need for immunosuppression, because we don't want to have to give somebody a different challenge just to solve their diabetes. We don't want to leave them without an immune system. And a scarce supply of donors. There's just not that many deceased donor islet cells available to really scale up a treatment. Now, in the last 50 years, there's been some terrific progress made, in particular around the scarce donor supply. Today, there are numerous companies that have protocols around iPSC-derived islets that basically represent an opportunity for an unlimited supply of those cells. But the failed engraftment and the need for immunosuppression still represent areas that need significant improvement. There are not good solutions for that today. So I'm here to talk to you about what we're doing at Fluidform. And what we're doing at Fluidform is really focused on this idea that we can eliminate insulin injections to cure type 1 diabetes. This has been a focus of ours now. We are really, really excited about what we'll share with you. We believe that the most important thing here is that it starts with that core belief that the idea that cell therapy can cure type 1 diabetes is very well established. If we can get the cells in the right kind of environment where we can avoid systemic immune suppression, we don't have to implant in vital tissue and organs, and we can keep those cells alive, we know that this is a solvable problem. The challenge has been that today there simply aren't solutions that tick all 3 of those boxes. And so when we sat down to work on this as a program here at Fluidform, our first question was, what if we could tick all 3? And what that looks like is an implant that goes just underneath the skin. It goes just underneath the skin so that it can be easily removed if it's needed. It's not implanted in the liver or in the kidney where removing it would potentially risk those organs. It's an implant that doesn't require systemic immune suppression because it carries a local dose of immune suppression just for the area that's needed and not for the rest of the body. And because of our proprietary technology, we can furnish that implant with a lot of blood supply right underneath the skin. We don't have to go deep in the body to the liver or the kidney or other locations to get the cells the blood supply they need. So this is sort of that important trinity: minimally invasive, easily retrievable, highly vascularized. We can solve the problem with a very, very different approach to what everybody else is doing today. What does it look like? What does that implant look like? It's really made up of about 3 ingredients on the inside. These light pink circles that you see there, those represent the insulin-producing beta cells. They come from stem cells. They produce insulin just like the cells in your pancreas, and our construct is loaded with them. They're surrounded by what are called extracellular matrix proteins. These are things that you might have heard of, like collagen or fibrin. And these are the proteins that surround cells in their native environments that tell cells, give the cells the signals that they are okay. They are in an environment where it's normal, natural, healthy for them to behave the way they would if they were inside of a pancreas. The third ingredient, cartooned here by these green circles, reflects a local depot of an immunosuppressant drug that's designed to sort of diffuse throughout this implant without then creating a high level anywhere else in the body. It offers that local protection just in the area where we need it, and it doesn't come with all of the side effects of systemic immune suppression. Now, the other thing that you'll see in this cartoon is the vasculature, the blood vessels that are growing into the tissue. And that is an effect that we are able to leverage because our technology platform, when we build these tissues, we are able to promote vascular ingrowth really, really quickly in the area just underneath the skin. And that's what we'll talk to you about next. So our technology, which is a technology called 3D bioprinting, it allows us to use a 3D printer, robotic assembly, repeatable, reliable, in order to deposit cells and proteins and help them coalesce into an actual tissue. In this case here, we think about it as a mini pancreas tissue. Our technology starts by 3D printing those materials inside of a special gel matrix. It takes just a couple of minutes. And that gel matrix is really important because it allows us to control things like the porosity of what we print, like the biochemistry of what we print. And those levels of control are what lets us drive that effect of that vascularization deep inside. What it looks like is cartooned out here. This is an animation of a 3D bioprint using our fresh technology, where you can see that the materials are deposited inside of that gel. They're left to do the self-assembly that they know how to do really, really well on their own. And then the gel is washed away at body temperature, and what's left behind is that 3D printed tissue. Now, what's really exciting about that is when we implant that tissue underneath the skin, we see enormous amounts of new blood vessel growth. And it happens very quickly. The data that you're looking at here was published in Science in 2019, and it compares 2 discs that were implanted underneath the skin, both made of exactly the same materials in exactly the same amounts. One disc was casted, just poured into a dish and allowed to turn into a disc. The other one was fresh printed. And you can see on the right here, that fresh printed disc is much redder. Just as a quick examination, you can tell that that has a lot more blood supply. And when we zoom in with microscopy, we see blood supply everywhere throughout there, all the way down to a capillary scale. That's really important because it's the capillaries that actually nourish the cells by providing them oxygen and giving them access to the blood glucose to know what to do. This is a fly-through of that upright microscopy that was done, and you can see it's not just one small area that has new blood vessel ingrowth, it's everywhere. All of these are stained to demonstrate that they're hooked up, in this case to the mouse that was implanted inside, to the mouse's native blood supply, and this has now been fully integrated. So what we do when we build these pancreas tissues is we leverage this effect, the ability to drive vascular ingrowth, in order to make these pancreas cell implants just underneath the skin in an easily retrievable location. The way we demonstrate that that works is we actually take an immune compromised mouse, a mouse without an immune system, and we give it an injection that causes basically a version of diabetes. It causes its blood sugars to go up really quickly because the mouse is no longer able to produce its own insulin. On the day zero, that sort of dotted vertical line on this chart, then we implanted either our scaffold with the insulin producing human islet cells inside of it, or the same amount of islet cells, but not printed into our scaffold. So the R scaffold is the blue line, the same amount of islet cells in the same location, just not printed into a scaffold, that's that gray line. And you can see a really big difference. When we print those scaffolds into a pancreas tissue, we're able to see a very rapid drop over the span of 2 to 3 weeks back down to a normal blood sugar level. And then it stays there. It stays there consistently and durably for 5, 6 months. We're really pleased to see just how effective that works, because these are mice with a fairly short lifespan. And so this is basically over the majority of this mouse's lifespan. These mice are free to roam, free to eat whatever they like. There's no special diet or anything else like that. We're able to see an extraordinary level of blood glucose control. Now compare and contrast that to the gray line. That's the same kind of mouse with the same kind of condition, implanted with the same number of insulin producing cells in the same location. The only difference is it was not printed using our technology, and therefore it doesn't get the benefit of the blood vessels that grow in that make those cells healthy and functional for a long time. And you can see you just don't get that level of blood glucose control. And eventually they're not able to last out that period of time because they're very unhealthy with such a high blood glucose level. So this is why we're so excited about the work that we're doing, because we're seeing the effect. It's real, it's durable, and it's incredibly promising. The other thing that we see that we're so excited about is when we take the scaffold out, we see an immediate jump right back up to that high blood sugar level. So we know it's the scaffold that we implanted that's doing the job. And now where we are is we are so excited to start the next round of studies that will both include some additional work in the mouse to build on the work that we've done, showing that we can do local control of the immune system, to demonstrate that in the mouse, and to do the first studies in a larger animal. In our case, we'll be doing those studies in a pig. And that's the next step towards the translation that we'd like to do to eventually get this treatment into humans. So with that, I'm going to pause. I'm going to say thank you. And I'm going to switch back over to, oh, there we go, to take a look at the Q&A and see what questions we might have. At this moment, if you do have any questions, please do feel free to use the Q&A function. It should be up at the top of your screen. I'll make sure we've got questions that are populated here, and happy to answer any questions that come up. Yeah, the first question that's come up is, how long do we expect it's going to be before we can get into human trials? And that's a good question. We are at the moment expecting to be doing work that's going to take us between this year and next year to develop all the data that we need to submit to the FDA. At that time, then we expect to go through the process to get an approval to run a trial. And our hope is to be able to be in human trials in 2027. That is definitely, there's a lot of things that we need to have go right in that situation. And so there's a lot of uncertainty around exactly how quickly those things can happen. But our belief is that we can get there. We can get there with some really high quality data, and that we can be on a process, on the path to be treating humans before the end of 27. We had a couple other questions that came in beforehand. The second question that came in was really around the immunosuppression. So we talked about a local immune suppression, and how is that different from the immune suppression that's being used today in the trials that are ongoing. So for example, Vertex Pharmaceuticals has a trial that relies on immunosuppression. And that immunosuppression that Vertex relies on basically knocks out the immune system throughout the whole body. And so that is there are a couple of different drugs in there, tacrolimus, rapamycin. They're really designed for an anti-rejection of an organ transplant as a primary purpose. Our system uses some of those same kinds of drugs, but uses them locally, just in the area where the tissue is, and uses about 100 to 1,000-fold less drug to achieve the same effect because we only need to protect that one small area. Because we use so much less drug, there's vastly less drug throughout the rest of the body, which means that we don't expect to see any of the same kinds of side effects of chronic immune suppression systemically. So a question that just came in from Chris, thank you, is around what we expect to do with the additional funds that can accelerate our work. That's a great question. Thank you, Chris. So one of the things that we're so excited to do is to be taking some of the studies that we have planned for later this year and early next year and being able to start them quickly. Right. So what we have coming up now is a series of a couple of studies. One of the studies involves doing the work that we've done historically with human donor cadaver islet cells and doing those with cells that come from stem cells. We've got a really exciting partnership on the table with a very advanced line of these human stem cells, and we are advancing some work right now in the mouse. We are really excited to take that and bring it up to the next level. And the most important thing that we're really looking forward to is getting the studies started in the porcine model, in the pig model. Being able to go from the small animal testing up to the large animal testing, we think will be one of the biggest accelerants that we can have to being able to collect the data that's going to be important to the FDA. Thank you for that. All right, I'll just give it one more minute in case there's any other questions that come up on the line. And in the meantime, I'd just like to thank you all so much for attending. It's really great to have your curiosity and your interest in the company. We're really excited to share with you what we're doing. And as any other questions come up, through the Start Engine portal, please don't hesitate. There's a great opportunity to ask questions there. And the benefit of that is if you have that question, it's entirely possible other people do too. And that's a great way to get that question out. We can get that answered, and that way everybody can get the benefit of learning from that. All right, well, with that, we'll bring the webinar to a close. Thank you so much for attending, and we look forward to any other questions or any other feedback that you might have. I hope you have a great day.